Thanks. I'd like to thank the organizers again for inviting us to be part of Eyecelerator. As we are a publicly traded company, these are some of our legal disclaimers. So at Surrozen, we're focused on developing multifunctional antibodies for retinal vascular diseases. And I think we and others are really focused on the therapeutic hypothesis that combining multiple mechanisms with VEGF are really needed to transcend the standard of care and meet the unmet needs for those with retinal vascular diseases. And at Surrozen, we're focused on three clinically validated pathways listed above, of the Wnt pathway, which we just heard about, as well as VEGF and interleukin-6. And we're coming about it from the standpoint of deep expertise in Wnt biology and antibody engineering. We were started over 10 years ago, really by the pioneers in Wnt signaling, and have published and made a lot of the seminal insights to develop this new class of therapeutics. And we're now applying this platform in combination with other drivers focused in ophthalmology. And I'll talk a little bit about our pipeline and some of our combination assets, which combine Wnt agonism with VEGF inhibition and/or IL-6. And our lead asset will be in the clinic later this year. So first, a little bit more about the Wnt pathway. My colleague did a great job of introducing it in the previous slide. But this pathway is really a key regulator of both retinal vascular development as well as maintenance. And here we're going to talk about some of the key players again. Those are Frizzled-4 and LRP5 and Norrin. And so Norrin is the Wnt ligand that binds Frizzled-4 and LRP5. And so these are Wnt receptors on the surface of retinal endothelial cells to provide these therapeutic activities of maintaining the blood-retina barrier and promoting vascular repair and reperfusion. And why do we know that these specifically proteins are important? Well, one evidence is from human genetics. So mutations in each of these genes lead to incomplete development of the vasculature in humans, leading to ischemia and vision loss. There's also preclinical data that if we were to activate this pathway through binding of Frizzled-4 and LRP5, that this is able to both restore vascular integrity, reduce leakage, as well as, remarkably, even revascularize areas of absent vessels in preclinical models. And now there's also clinical validation with the first generation of Wnt agonist, which binds Frizzled-4 and LRP5, showing promising results in an early stage DME study and is now fully enrolled in phase 3 studies. And at Surrozen, we're focused on, again, marrying these two mechanisms together, and that's reflected in our lead molecule, SCN-8141. And so you can see here the dual mechanisms of Wnt activation through LRP5 and Frizzled-4 binding elements, as well as VEGF inhibition elements on the other side. And one question you might ask is, well, does really combining these mechanisms lead to additive or synergistic efficacy? And I'd like to show one piece of clinical data in which we tried to address this question. And so what we did is we took our SCN-8141 molecule and then systematically either eliminated the Wnt activity through point mutations or the anti-VEGF activity through point mutations, and then tested each of those serially in the oxygen-induced retinopathy model, as well as a combination of those two. You can think of it as a co-formulation with our single molecule. And these were all given at the same equimolar dose. And what you can see on both of the metrics in this model, both the reduction of non-perfusion as well as the reduction of neovascularization, is that our single molecule had superior efficacy compared to either of the monotherapies, but even surprisingly compared to the combination together, suggesting there might be some synergy by combining these in the same molecule. And we may think this may translate to greater clinical efficacy and durability. And we have a poster on Monday with this data and others, so I encourage you to come by. And so I'll end with our pipeline. As I mentioned with our asset SCN-8141, we're very excited to move that into the clinic later this year. And then following on that, we also have another molecule, SCN-8143. So that's a trifunctional antibody, which adds IL-6 inhibition on top of the aforementioned Wnt activation and VEGF inhibition. We also have our original asset that we've developed in retinopathies. This is a Frizzled-4 LRP5 Wnt agonist, that's been licensed and is in development by our partner, Boehringer Ingelheim. In addition, we have a brewing and very vigorous research pipeline looking at additional Wnt receptors with some very promising data both in Fuchs endothelial corneal dystrophy as well as geographic atrophy. So I want to thank you for your attention.